Assalamu alaikum, everyone. My name is Aduli Khan, an associate of NSI GMSEP. I'll give you a brief introduction. First of all, you are all welcome to our workshop. We have a distinguished speaker, Dr. Farooq, and Dr. Abel here. Dr. Abel would introduce the neuroanatomy lecture, and then Dr. Farooq would do the hands-on practices. So for starters, I'll give some of an introduction of our society. NSIG, Neuroscience Interest Group, is a part of Medical Student Association, Pakistan, which specializes in multi-institutional organization, जहाँ पे उसकी different branches हैं. हमारे पास research branch है, clinical training branch है, और ये neuroscience interest group हमारी personal branch है जो आपके लिए brain dissection workshop लाई है in collaboration with Mission Brain from UMDC, which is also a society, which is also an organization which does different things. Now, before starting, I would like to call Arkam Siddiqui so that he can recite the Holy Quran with us.
أعوذ بالله من الشيطان الرجيم. بسم الله الرحمن الرحيم. لا يكلف الله نفسا إلا وسعها لها ما كسبت وعليها ما اكتسبت ربنا لا تؤاخذنا إن نسينا أو أخطأنا ربنا ولا تحمل علينا إصرا كما حملته على الذين من قبلنا ربنا ولا تحملنا ما لا طاقة لنا به واعف عنا واغفر لنا وارحمنا أنت مولانا فانصرنا على القوم الكافرين صدق الله العظيم.
Subhanallah. Now, I think we'll start the workshop. I would like to call Dr. Abel for the neuroanatomy lecture for us.
Assalamu alaikum. Can you hear me? Assalamu alaikum. My name is Dr. Abel. I'm assistant professor of neurosurgery here in this department, and I welcome all of you for this workshop, this really good workshop which is being organized for brain dissection. And the purpose of this workshop is basically to let you give a chance to move on from textbook anatomy and then experience the real anatomy on the brain, right? So we'll start with a very basic, very simple neuroanatomy presentation, so that you know what you want when we move on to the practical dissections. You know what to see, what to look for, and what are the critical structures that you should know once we start the workshop. So first, can you tell me which majority of the audience here is like first year to final year? Everybody, all the years are included in this workshop. So I'll just keep it a little bit simple for everyone. Second year forwards, second year till final year. Okay, so what? Do we have the, so that I can flip? So the learning objectives in this workshop would be, in this lecture basically would be to understand the basic neuroanatomy of what we're seeing on the surface of the brain and what we're seeing inside the brain. So we'll first talk simply about what are the important structural elements that you should know about when we start the brain dissection, and what we will see when we move from outwards to inwards, right? So the surface of the brain is composed of the cerebral cortex, right? So which is approximately 2 to 4 mm thick, and which is basically the main. Okay. So Okay, so basically the cortex is the main critical structure which makes the brain what it is, right? So it is the uppermost layer of the brain, approximately 2 to 4 mm thick, and it contains the cell bodies. ये कनेक्ट नहीं हो रहा उस पर, तो लाइक लैपटॉप पे है ना, नहीं ये इसमें कनेक्ट करना होगा ना यूएसबी, तो ये चल जाएगा, ठीक है, लैपटॉप पे डायरेक्ट. Okay, so this what you're seeing over here on the surface is the cortex, right? And cortex is very critical structure. जब हम surgery करते हैं, we want to make sure that we don't go into eloquent areas. What are eloquent areas? Which are areas of the brain which have critical functions like the motor area, the sensory motor cortex, the speech areas. And when we're dissecting the brain, we avoid these areas. So obviously we need to know the exact location of these areas and the landmarks on the surface which will help us. This model obviously has color codings, right? But when you're seeing the brain, you have no idea where the motor cortex is, where the other cortex, because everything looks the same, right? But we have some critical landmarks, and we identify these landmarks, and once we identify them, we know which areas to avoid, which are safe zones through which we can go for dissection, which are zones that we need to preserve at all costs, right? So on the surface of the brain, we have two cerebral hemispheres for both sides, the right side and the left side. The right side controls the left side of the body, the left side controls the right side of the body. In 70% of the humans, the left side is dominant, right? So we assume, some people say that like most of the times when we want to establish dominancy of the brain, we ask if the patient is right-handed or left-handed, right? And usually right-handed people are left side dominant, but it's not always true. Sometimes even left-handed people can also be left-sided dominant, right? So we generally assume that the left side is the dominant lobe. And what I mean by the dominant lobe is the majority of the function is in the left side of the brain, right? So speech, memory, and any damage to the left side of the brain would result in more devastating neurological deficits in a patient as compared to the right sided lesion, right? So first we have to identify which is the right side and the left side, right? So from your perspective, which do you think is the right side of the brain? Like when we're looking on the table and we're standing behind the patient, how would we know which is the right side? Look, we need to be oriented, right? So if you're looking at it directly, then you have to look at it differently. And when you're operating the patient, when you're dissecting the brain, you have to look at it differently, right? But you need to first identify the left and the right side. Both cerebral hemispheres are connected in between through the corpus callosum, right? So corpus callosum is the bridge between the two lobes. If you cut through the corpus callosum, what happens? Both lobes become independent of one another. The patient does not die, but he will become independent. The right side and the left side will have no means of communication with each other. So the corpus callosum must be preserved at all costs, right? And how do we know where the midline of the brain is? What is the landmark that you see on the surface? What is the name of this fissure? Further divided into how many parts?